our today's guest speaker, faculty members, and my dear students. On behalf of Ideal College of Pharmacy and Research, I, Suvarna Patil, take this opportunity to welcome our guest and all the participants to the live webinar on the extremely relevant topic of pharmacy profession. Today we have privilege to have expert from the field of pharmacy who shall be reflecting the theme of this webinar. Now I would like to introduce our today's guest and speaker, Mr. Yogesh Shirole sir. Sir did his M.Pharm in pharmaceutics from NH Hiranandani College of Pharmacy, Ulhasnagar, and he is currently pursuing PhD from NIMS University, Jaipur. He has 8 years of experience in pharma industry. He worked as an IPQ chemist in Vardhaman Export, Mumbai, and was involved in performing various IPQ activities and related documentation. He has a vast experience of formulation development for various solid oral dosage form and currently working as a research scientist at McLeod's Pharmaceutical as a research scientist in formulation development department. With this brief introduction, I request Mr. Yogesh Shirve sir to deliver his talk on new drug development, its FDA guidelines and commercialization. Over to you, sir.
Uh, Good afternoon to all of you. Thank you, ma'am, for introduction. Uh, Myself, Yogesh Rode. Today, we are going to start uh, information on the new drug development, its approval, and commercialization. Next, uh, next. Before start with the development, before start with the drug development, we should learn about a regulatory agency which hold and regulation and supervision of on the drug developments is Food and Drug Application. It is a federal agency developed by United States in 1906. FDA is responsible for protecting and promoting the public health through the regulation and supervision of food safety, dietary supplement, biopharmaceuticals, prescriptions and OTC pharmaceutical drugs and medical devices. Then next, we should know about active pharmaceutical ingredient. What is active pharmaceutical ingredient? It is a drug substance, is the API or component that produces the pharmacological activity. API may be and it is may be produced by many ways like chemical synthesis, recover from natural sources, then recombinant DNA technology, fermentation, or the combination of all these processes. Then, what is new chemical entity? It is a drug substance with the unknown clinical, toxicological, and physical and chemical properties. Then, uh, next, next is the drug product. A drug product is a finished dosage form which contains an API along with the other ingredient that may be a inert or we can call it as a excipient and this is developed into the finished dosage form like capsules, tablets, injectables, etc. Then next, what are the new drug development FDA guidelines? uh, Before in the development FDA guidelines, the drug is developed in and its approval takes place into a several steps. So these steps are as follows: preclinical testing, then phase 1, phase 2, phase 3, phase 4, and phase 5. So next is a preclinical testing. So what we call as preclinical testing. In this preclinical testing, the animal study or toxicological data is developed on and it is carried out on animals. In this, no attempt is made to develop a final formulation in this stage. So here we will just do the study on animals and generates data which can be used for further studies in phase 1, phase 2, phase 3. So next is phase 1. Before starting with the phase 1, we need to have some prerequisites or we need to file we need to have some the submissions, like we need to submit IND, investigational new drug application, and once it is approved, we can start with the phase 1 studies. Then we should have an consent from investigator and volunteers before starting of this stage. Here we are using the healthy volunteers in this stage for clinical studies, but the number of volunteers are less to generate the safety and to generate the dose, how much dose we can give, maximum dose we can give to patient. All these studies are carried out in phase 1 studies. Then next are phase 2 studies. So phase 2 studies are like here limited number of patients of data. So here we can see bioavailability studies and pharmacokinetics are measured in this phase. A drug formulation having a good physicochemical properties are also developed. So here we can monitor toxicity studies in this phase. So once the phase 2 is completed, the data can be useful for carrying out phase 3 studies. So in phase 3 studies, we can use more number of patients to generate the and to verify that effectiveness of the formulation. So once the phase 2 is completed, we can start with the phase 3. So next is phase 3. Here a large scale production of drug is carried out and shall be used for to monitor and verify the effectiveness of formulation, and also in this stage the side effects of the drugs are monitored. So objective of this study is to determine the safety and efficacy of drug product in large diseased human sample. So here persons with the disease are selected for further, and once these studies are getting completed, we can submit the formulation or dosage form to as a new NDA to FDA for their review and subsequent approval. Once FDA approval come, we can start with the phase 4 study. So FDA provides its approval on our NDA, new drug applications. So that the drug is satisfactory by all parameters and it's safe as demonstrated by our animal and human studies. Next is phase 4 studies. After new drug application submission and its approval, the large scale manufacturing of products gets start. Once we will get the approval on our NDA, we'll start the manufacturing of our large scale manufacturing of our drug product, and its packaging is carried on. So additional clinical studies we can calculate, we can start here to develop the additional clinical studies, and which can be performed on patients with the different group age like elderly, pediatrics and renal impaired patients. So after that NDA application will go with the phase 4 studies. After NDA submissions and its approval, we will start the uh, phase 4 studies. Next, phase 5 studies. Once in phase 5 studies, the slight changes in the drug formulations we can do, or that can be reported in correspond to the SUPAC guidelines. The change in the drug formulation and it should be comply with the SUPAC guidelines. So all these changes we can do in phase 4 drug development. The next is another is the product line extension, like only a or physical form or strength is changed in the same indication, like the dose is given for another in another form. Then developing the transdermal patch when only one tablets have been available, like for example nitroglycerin, then nicotine. Transdermal patches are it is available in tablets, and we can also develop a transdermal patches. And another is additional state. If the product has some one or two strains in line, and if you want to have an additional strain, it should be in the range of total maximum daily dose, and we can develop that additional strength and submit to FDA for their review in phase 4, phase 5. Then next is the biological products. So biological products according to FDA is an antibiotics, serum, toxins, vaccines, bloods or blood components or its derivatives. So allergenic products or analogs products are applicable to the prevention or treatment of the disease or injuries. So as we know the example of biological products like vaccines, etc., and other serums, then toxins, antitoxins, this all comes under biological products. So for this, we need to have an biological license application for marketing of these drugs under Public Health Service Act. So before going for this commercial distribution, we should have an BLA, biological license application. Then next, generic drug products. So here the difference comes. It's as I explained earlier, new drug product. So once the new drug application has been submitted to an FDA, it is get and once it is approved, will manufacture a drug for subsequent years. Once it goes under an patent filing and patent application, and will manufacture the drug for subsequent years till the patent expiry. Once the patent is about to expire, and other manufacturer can enter into the market, and this strategy is called, or this product is called as generic drug product. So other companies or other formulators can enter into a market once a patent is expired, and this development is called as generic drug development. So, as all the preclinicals and clinical studies are carried out satisfactorily in the development of new drugs, so here just we need to do an bioequivalence study, and we need to prove our generic product is bioequivalent with the existing available drug product. So here we have to carry out the bioequivalence studies only in the healthy volunteer. Then these generic products, uh, we have to make an compliance in each and every aspects with the regular reference listed drug and reference standard for the generic product, like its size, shape, color, or the amount of excipient we use, or we can change some sort of excipients in the development. But we need to develop an bioequivalence study before filing of these excipients, and we should have an sufficient stability data and safety data for the excipients which we are going to use in the development of generic products. Then next, before the generic product is marketed, the manufacturer will have to and submit and another application for agency for its approval. Like in new drug development, we are submitting an application is called as new drug application. Here, it is a generic product, we have to given an application called as ANDA. ANDA means abbreviated new drug application, means and abbreviated is an another application to existing new drug application. And since, as I mentioned earlier, since the NDA clinical trials have already been performed, here we have just to prove the bioequivalence studies for ANDA approval. Then next, then what are the another regulatory approvals for product line extension? Like for as I mentioned earlier, product line extensions are like change in form, physical form, or addition of new strength or addition of another dosage form. Here we need to develop an analytical records, some manufacturing records. Then we need to prove an stability studies in with respect to ICH guidelines uh, has been provided by an agency US FDA. So stability guidelines we have to follow, and we have to do the stability of our uh, new strength, and we have to compare with the uh, existing strength and its uh, reference product. So, and again, we have to develop the viability studies, then bioequivalence studies, safety studies, etc. for our new product line extension approval. So, then next is the FDA guidelines, that is pre-approval inspection. So, pre-approval inspection is the inspection carried out by FDA before before commercial manufacturing or before providing the approval for our drug. Once we submit the new drug applications or our as a ANDA applications for approval, the pre-approval inspection is carried out by an FDA agency here, and it is pre-approval inspection is required, like in case of major change and application to the NDA or ANDA. Next, there is pre-approval inspection. During the pre-approval inspection, FDA investigator investigate the drug development process, then drug development facility, then drug analysis facility, where all these states are being carried out, like its development facility where it is, then what is its manufacturing facility, then analytical facility. All these inspections are being carried out by FDA investigator along with the appropriate record, development records, then manufacturing record, analytical records are being verified by an investigator. Once it is get satisfied investigation, then he will submit the report to the agency, and after satisfactory submission of this report, agency will provide an approval for your manufacturing of the drugs under the subsequent categories like new drug applications, ANDA applications, AADA applications, and which will be used for to manufacture the product. Next, then scale up and post approval changes. So once the drug product is approved, we can commercialize or we can manufacture it in a bulk. So before manufacturing, we need to scale up that factor. So here in while scale up of the drug product, there we need to have a slight change in the manufacturing process, or we need to slightly change the excipients ratios in the manufacture drug product. So that change, uh, slight change we can rectify through a SUPAC guidelines. Here a SUPAC guidelines are these, all these changes are derived into the three phases, like level 1 change, level 2 change, level 3 changes. So here in level 1 changes, the change what we have done should not have an impact on the product quality. All the changes are categorized as level 1 changes, and the level 2 changes are the changes which have a slight impact on the product quality, are categorized into the level 2 changes. The level 3 changes are the changes which have an impact on the product quality and its efficacy, are being categorized into a level 3 changes. And all these categorized changes are being submitted to a agency for further approval, and once approvals are taken, we can manufacture the drug with the change in the SUPAC guideline. So if the SUPAC guideline is applicable for changing the site of manufacture or scale up or scale down the batch size of the formulation. Next, then the product withdrawal from the market. If any serious adverse effect is being noted or reported, then we can withdraw a product from, and we can apply to an agency and we can withdraw a product from market. The agency will recall the product from market. So here is the black box is warning for serious adverse effect, and this box warning is even mandatory to be mentioned in the leaflet of drug pack. So all this. Then the last is the table which summarize the different stages and approval of the drug development in table. So here in preclinical, it is carried out on animals and other toxicological data is being studied. Then phase 1, phase 2, phase 3 studies are carried out on healthy and human volunteers with the change in number of populations in each phase. In phase first, only 20 to 30 populations are being studied. In phase 2, several hundred populations are being studied for safety and effectiveness of the drug, and in phase 3 the safety and effectiveness of the drug which we have generated is verified in phase 3 with the and also monitor any adverse effect. So from phase 1 to phase 3 the investigational new drug applications are being uh, changed, like approximately 70% INDs are being transferred into phase 2. In phase 2 approximately 33% are being transferred into phase 3. In phase 3 around 24% are being transferred into the phase 4, and in phase 4 we are carried out these studies on large population with the bulk manufacturing and using the final dosage form, and in phase 5 we'll carry out the its bulk manufacturing and monitor its effectiveness on the dosage form. So this is all in short for drug development from its initial to its approval and further for its commercialization. Thank you. Any questions?
Hello, sir. Thank you so much, sir,
Hello.
for giving your valuable time. And I would like to ask one question, sir. So,
Yes,
like,
sir.
what is the difference between like branded drug and generic drug?
As I mentioned earlier, branded drugs, these are the means the drugs which comes under the patent category. The for like in new drug applications, we are submitting the new drug application to the agency, and the drugs which categorize under this category are called as branded drugs. Then generic drugs are the additional or abbreviated. It comes under the abbreviated new drug application. Once the patent gets expired, we can manufacture an generic drug for the existing available drug. So this is the difference between branded drugs and generic drugs.
Okay, sir. And one more question, sir. Sir, how can we evaluate safety and efficacy of new developed drug in any phase trial?
The safety and efficacy of drug in phase 3 trial we can develop, like in phase 3 we need to carry out a phase 3 on a several hundred population, and here in this population the investigator and agency use and technique called randomization. In like in randomization means random sampling. In all these the drug is given randomly to patients, like both our reference drug also and our test products also, and the effects are being monitored. Since the technique is randomization and double blind, here investigator is unknown of which patient has got the uh, this reference drug or our drug. So the results which we have got in this are very appropriate and can be extrapolated for its safety and effectiveness.
Okay, sir. And one more thing, sir. Our students also would like to know about uh, F FND. like R&D and F&D. Can you explain a few things regarding F&D, as you are working in F&D as a research scientist? So our student also would like to know few things about F&D.
Yeah, sure. Uh, since F&D it is a branch of R&D. So first comes the R&D, research and development. In the research and development, there are subsequent branches like F&D, A&D, PRD, packaging development. So the head branch is the R&D, research and development, and the sub branches are F&D, formulation development, then A&D, analytical development, then PRD, packaging development. So what comes under the F&D, formulation development? Here, like as I have mentioned in my webinar, for abbreviated new drug application. So in F&D, formulation and development, we are filing for an ANDA application for existing branded drug. We'll file our generic drug product and we'll prove the bioequivalence study and will file for the ANDA application. So the drug development carried out in several steps like uh, pre-steps, then prototype formulation, followed by its pilot bio-study, then its scale-up process, then its manufacturing, etc. And we need to support in each and aspects from its initial development to for till its commercial launch for drug. So F&D is a core branch which supports who supports for drug manufacturing and etc.
Okay. Thank you, sir. Thank you so much for giving your valuable time for deliver such a informative and innovative things you have delivered in this webinar. And this, whatever the knowledge you have given in this today's webinar, it will be definitely beneficial for our students, as our final year student will come as new graduates and they will go for industrial job. That time, this information will definitely beneficial for their bright future. Thank you so much, sir, for your valuable time and to deliver such an informative session for our student and our faculty member as well. And I would like to once again thank to Mr. Yogesh Shirore, sir, who's working as a research scientist in Macleods and there in Mumbai. Thank you, sir. And I would like to thank my all my faculty members who support me to organize this event. I also thankful for my principal ma'am who gave permission to arrange this event. And thank you each and everyone. And at the last, thank you each and everyone who participate in this session. Thank you so much. And I declare the conclusion of this session. Thank you so much.
Thank you. Thank you, sir, for inviting.
Yes, sir. Thank you.
Thank you.